I'm Reason, one of the founders of Repair Biotechnologies, one of the companies in the new longevity industry. I thought I'd talk today about our atherosclerosis program, one of our two programs. So I believe I can probably skip right over all of the interesting stuff about how the aging market is going to be enormous. Everybody here should be well aware of that, and just start with who we are. So we're a preclinical biotechnology company. We started in 2018 and really got going in 2019. We're developing a first-in-class therapy with the aim of reversing atherosclerosis and certain types of hypercholesterolemias. This works by providing cells with the ability to degrade cholesterol and oxidize cholesterol, the latter of which is a real problem as one gets older. So we're in the midst of preclinical development with animal studies. We have good cell data to point to leading into those. And if everything goes well, then we believe phase 1 is a 2022 sort of a time frame. And we hope to be able to hit that goal. So in order to understand why what we're doing is really interesting, one has to understand a little bit about atherosclerosis. Beyond the fact that it's a condition that kills between a tenth and a quarter of everybody, depending on how you want to count the numbers. So I think most people are aware that atherosclerosis is in some way connected to cholesterol, and that your blood vessels get gunked up and narrow and weaken, and eventually they explode and you either get a heart attack or a stroke as a consequence. So cholesterol is really sort of a byproduct of this whole business. The real issue with atherosclerosis is that macrophages, immune cells, responsible for keeping your arteries clean of all this gunk, really cannot deal with oxidized cholesterol. And oxidized cholesterol levels increase quite dramatically as you age. So in youth, this whole system works great. Your macrophages sweep up the cholesterol and hand it back into the bloodstream to be taken back to the liver. But when you are old, the macrophages become impacted by cholesterol. They become dysfunctional. They become what are known as foam cells, which are packed with little cholesterol droplets they can't deal with. They become inflammatory and cause chronic inflammation. And eventually they die and add their corpses to these growing lesions that are weakening and destroying your arteries. It's not a good thing. So the present state of the market for therapies is really focused almost extensively on reducing blood cholesterol. And this works a little. Those numbers on the bottom left there, you can see these are really not great numbers. These are live a couple of years longer and cancer will get you, but this disease would kill you in the end. And it doesn't stop it. It just slows it modestly. And the reason it slows it modestly is that if you reduce all cholesterol in the bloodstream, you're reducing oxidized cholesterol, and it gives your macrophages a little bit more of a breathing room to actually deal with this issue. We can do better than this, and we need to do better than this. The way to do better is to actually reverse the lesions, to tear them down, to let the tissues regenerate. And of course, if you can break down cholesterol and oxidize cholesterol, then hopefully you can let cells and macrophages get back to their jobs of fixing things up as they did in youth. So how are we doing this? So we have a technology that we licensed that's a collection of enzymes, a couple of different packages thereof, that convert cholesterol or break it down. And then if you deliver this to cells via some form of gene therapy, the cells then become capable of degrading cholesterol in ways that they could not naturally. And the paper there it covers the basis of this technology and some of the options. It's a little bit of a dry read, but it's very interesting if you're into protein engineering. So the first approach we take in this is to target these therapies to macrophage cells themselves, rather than just delivering them systemically, which is maybe also an option. So if we can get the macrophages to eat cholesterol, and particularly eat the oxidized cholesterol that normally causes them great harm, then in theory they should just get on with their jobs and you will never get atherosclerosis, and existing lesions will be dismantled gradually as the macrophages do their work. So we will get to testing this, and I will show you what we have done. This is data from the scientific team that originally produced this in cell studies, showing that, yes, you can get cells to express these enzymes, and they work quite well. They convert cholesterol into things that are not cholesterol, and the cells are pretty happy. It doesn't cause any real issues. The next step here, which we carried out, is to take a bunch of macrophages, put them in a dish, expose them to oxidized cholesterol, such as the oxidized LDL cholesterol used here. As you can see on the left, it makes them quite unhappy. The green is the foam droplet, the droplets of cholesterol in foam cells. So a substantial portion of this dish got converted into foam cells, and some died. But on the right, cells that express our enzyme package here in the red, they're fine. They're quite happy. They can sort their lives out. They're resistant to this. This is much more clear in this diagram, which is for 7-keto-cholesterol, which is a much worse a type of oxidized cholesterol. And as you can see, it just uniformly kills or turns into foam cells the macrophages in the dish there. But when we have macrophages that are treated and express the protein, they're completely resilient to this. So you don't see near any foam cells in there, and the ones that are foam cells are the ones that seem to have escaped the treatment. So this is a really good demonstration that this works. It's a good thing to do. So we are in parallel developing the vector that allows us to deliver this to tissues. We have this sort of evidence. This was a green fluorescent protein type of study in which we show that we can get a green fluorescent protein via our vector expressing in the macrophages that we care about, and the monocytes, which are precursors to macrophages. About half of the monocytes in the body exist in the spleen at any one point in time. So you want to be able to get your therapy into the spleen if you administer it systemically. The other ones, you want to catch the ones that are actually in the blood vessel walls. So we looked at the aorta, and yep, it all seems good. So this is one of the fundamentals of our delivery technology that we're working on right now. Of note, earlier this year, we conducted an experiment in which we delivered this technology without any sort of restriction to cell type into mice using an AAV viral vector. Now, of course, if you do this, most of your vector goes to the lungs and the liver. We chose to look at the liver. And we found that we dramatically reduced cholesterol in the liver in a matter of a couple of weeks. This is a really powerful result. It's a very large effect size and done quite quickly. Now, the really interesting thing is not that we did this and achieved this result. The really interesting thing is that the mice appeared fine. So their serum cholesterol didn't really change at all. And their weights were no different between the controls who were untreated and the various groups who were treated with different variants of this approach. So the mice no doubt had side effects, but they weren't so serious as to rule this out as an approach. And when you can deliver something systemically in this way with no restriction, it opens up all sorts of doors in terms of potential conditions that can be treated without too much optimization of the therapy beyond where it stands today. And this slide just demonstrates that we can, we did do the expression here. In that study, we checked the liver tissue, and yes, the therapeutic proteins are very much expressed there. Okay, so in terms of IP, we've licensed this technology from academia with a very broad patent, and we are adding on top of that with our own patents as we demonstrate things such as the study results I just showed you, yes, it does interesting things in various tissues. Therefore, it's potentially applicable to all sorts of conditions involving too much cholesterol in those tissues. There's a range of genetic conditions, inherited conditions, and others in which there is just too much cholesterol in some part of the body and it's causing issues. In terms of where we're going, first off, I mean, obviously atherosclerosis is the real target here. It's the thing that kills enormous numbers of people. But it's not the first place we will try on the grounds that atherosclerosis studies are tremendously costly. So there's a condition called homozygous familial hypercholesterolemia. Note that I've had 2 years in which to learn how to say that quickly, multiple times. People who have this condition suffer from a genetic mutation that effectively causes them to undergo accelerated atherosclerosis. They have enormously huge levels of blood cholesterol. Interestingly, this causes no other effects beyond the accelerated atherosclerosis. It is evolutionarily very interesting that humans seem to be able to thrive with a tenth of the normal level of cholesterol, or 10 times the normal level of cholesterol, with no effect other than how fast are you going to develop atherosclerosis. It's very, very interesting that this can be so variable without more severe problems manifesting. So this condition is really unpleasant. People die at age 30 from atherosclerosis if they're untreated. They tend to be discovered late with significant lesions. So a way to reverse lesions for this population would be tremendously useful, because just the technologies that reduce blood cholesterol dramatically do not really help them reverse the lesions they have. So after doing that, we will then go on to the range of other conditions we can potentially treat with this technology. So the team, myself, Reason, and Bill Sherman, we founded the company. Bill Gannon keeps us on the straight and narrow. Mark is our principal scientist at the moment. And we have the adult supervision of the fellow who invented this technology, Richard Honcannon, and a number of other folk who keep us on the straight and narrow in terms of telling us how to mount various studies that we need to study, and so on and so forth. So I hope that as we move forward, we will be able to share with you all many more interesting results, such as the one I showed today, and we will carry forward. And the grand hope here is that we can really do something about this enormous killer of many people, atherosclerosis, by just trying a new approach here, something that has not been attempted before but seems very plausible as a way to actually reverse the harm that's done by this condition. Thank you.